So let's talk about a few questions of National Testing OVAS app in the TEST-25 biology discussion. So first question. So in meiosis, crossing over is initiated at, we know that meiosis is divided into two, meiosis 1 and meiosis 2. Meiosis 1 is also divided into prophase 1, metaphase 1, anaphase 1 and telophase 1, right? Now meiosis prophase 1 have several steps are there, like leptotene, zygote, pachytene, diplotin and diakinesis. Now crossing over initiated at crossing over is occurs in pachytene stage of the meiosis prophase 1, the pachytene stage, and leptotene stage, during leptotene stage bouquet stage, bouquet stage form in the during leptotene stage, and zygote stage the synaptonemal complex, synaptonemal complex form, and in during diplotin stage chiasma formation. These are major important steps that occur during the meiosis prophase 1. So answer will be the option A is the correct answer. Now next question. Most of the fungi are heterotrophic because they are, most of the fungi are belongs to all groups of fungi are saprotrophics. They are saprotrophics. They take their food from dead decaying organic matters. Fungi are parasitic like Puccinia, Gramineous tritrici. This is also parasite, and symbiotic. Fungi also in symbiotic association with lichen. Case of lichens, we know that lichen is the association between algae and fungus. Algae and fungus is the mutualistic association between algae and fungus. So algae also have plus plus interaction, both get benefited from each other. And another is the mycorrhiza also symbiotic association, fungus and the plant root, fungi and plant root. Mycorrhiza also divided into two types, ectomycorrhiza and endomycorrhiza. Ectomycorrhiza and endomycorrhiza. So correct answers is all of these are the correct answers. So next question. Now, which of the following hormones can play a significant role in osteoporosis? We know that osteoporosis is the breakdown of osteoporosis is the breakdown of or degradation of bones. That's osteoporosis. So osteoporosis basically, most of the important is the estrogen play important role in osteoporosis. Estrogen is a steroid or sex hormone that is essential to female bone health because it promotes the activity of osteoblasts, which are the cells that produce bone. When estrogen level drops during menopause, the osteoblasts are not able to effectively produce bone. So estrogen play important role. Another hormone is the parathyroid hormones. Parathyroid hormones also play important roles in calcium retention. So it's a parathormones is the antagonist of calcitonin. Parathormone also antagonist of calcitonin. So correct answer is estrogen and parathyroid hormones. So in case of aldosterone is not, aldosterone is from aldosterone is not. So another is the parathyroid. Prolactin also not involved in osteoporosis. So answer is option A. And we know that at so next question, the translation during translocation of organic solute in sieve tube member is supported by translocation in sieve tube membrane, sieve tube members. So during translocation, suppose these are mesophyll cells. These are mesophyll cells from where the photosynthate. During photosynthate are produced in the forms of the glucose molecule, and it transported in the forms of sucrose molecule. As the sucrose is non-reducing sugar, sucrose is less reactive. That's why during transport, plant transport in sucrose form, not in the glucose form. Now we can, these are mesophyll cells, mesophyll cells. Now mm, here, now these are from mesophyll cell, the photosynthate that is transported into the bundle seed cell, bundle seed cell. Now from bundle seed cell, it will be moved into the companion cells. Now here is the seed tube element, seed tube. Now here, these are our companion cells. This is also known as the transfer cell, companion cell, companion cells. Now from here, the sucrose is a sucrose molecule. Now sucrose are transported here due to concentration, less concentration gradient by diffusion. It's transported. Now here, that sucrose H plus importer is there. Here sucrose H plus importer in presence of utilizing ATP converted into ADP, and it will transport along with H plus ion. It will also transport the sucrose molecule. So sucrose molecule will reach here due to the presence of sucrose H plus importer, ATP converted into ADP by using proton and sucrose. They together will transport here, so sucrose will come here. So sucrose in that way, sucrose reach into the from companion, from mesophyll cells, then bundle seed cell. From bundle seed cell, it will reach into the companion cell in the presence of sucrose H plus importer. Another is that the P protein. P protein actually, now look at the option. The translocation of organic solute in seed tube member is supported by P protein. Actually, it's a not damage repairing protein. If it is a seed tube, any minor damage is there in the seed tube, and then P protein is involved. Another is the callose plug formation. Callose plug formation. If the damage is higher, then callose plug formation occurs. Callose is a calcium dependent, and so callose plug formation, it's a permanent and or large damage if occurs, then the callose plug formation occurs. If the smaller damage is there, then P protein is involved. And now cytoplasmic streaming, no, cytoplasmic streaming for transport is not there, and root pressure and transpiration pool for these are not for xylem transport, for water molecule transport is involved. So these are also not for. So most applicable is the mass flow hypothesis involving a carrier and a ATP. Mass flow hypothesis involving a carrier and a ATP. So next question. Now, which of the following pairs of substances are found inside a mitochondrion? Now, we know that mitochondria is a semi-autonomous organelle Mitochondria. Why mitochondria is called semi-autonomous organelle? Because mitochondria contain its own DNA and ribosome. Mitochondria have own DNA and its own ribosome. Mitochondria, we know that the mitochondria is have a double-stranded circular DNA molecules. It's have own double-stranded single DNA molecule, double stranded single DNA molecules. Another is that 70S type of ribosome is there. So that's why it's called endosymbiotic origin. Bacteria from a mitochondria actually once upon a time it was a free living bacteria, and that's now it remain in our eukaryotic cells. This theory is called endosymbiotic theory. Endosymbiotic theory. So endosymbiotic theory support that this also like bacteria 70s prokaryotic ribosome is there. So that's a single DNA molecules and a ribosome. RNA is not there. It's a single DNA molecule 50s, not single RNA molecule. So answer is the option D is the correct answer. So next is one of the effect of gibberellin is to induce the synthesis of amylase enzyme which cause the breakdown of starch and provide the nourishment to the embryo in the seed. This phenomenon leads to germination of seed. We know that in the gibberellin is involved in seed germination by increasing the activity of the alpha amylase in the aleuron layer. Activity in the alpha amylase activity increases alpha amylase activity. Now, antagonist are say opposite in function, means they are actually the opposite function of gibberellin is the antagonist to this phenomena. Antagonist and agonist. Antagonist which actually opposite in function. Gibberellin. So gibberellin opposite in function is auxin is also promoting growth promoting, kinetin, cytokinin, so also growth promoting. So ethylene also in ethylene involved in cyanation, and ethylene and so most of the appropriate is the abscisic acid or AVA is the correct answer. Now, which of the following is not a characteristic features of sponge? We know that sponge belong to poriferans. Poriferans and it's have a pore bearing animals. Poriferans are pore bearing animals. So pore bearing animals they have larger this they have their body contain multiple pores. The small pores are called ostia and the larger pore is known as osculum. Suppose this is now this large pore is called osculum and the smaller pores are called ostia. So the presence of ostia is the major one. They also so presence of ostia is the character of poriferans, and they have the cellular level of organization, and they are digestion. Their digestion also they are several cells are there like guanocyte cells, pinacocyte cells are there. Guanocytes are flagellated. They actually due to the water current, the food particle enters, and its digestion is also intracellular digestion occurs and the body supported by chitin. The body is not supported by body is calcareous. It's a calcium carbonate. So chitin is a polysaccharide, and it's a chitin is only present in only on the most of the fungus cell wall. Fungi cell wall is made up of chitin. Another is the exoskeleton of arthropod. Exoskeleton of arthropod contain chitin. So chitin actually protective covering. Chitin only present in two group that is fungus, exoskeleton of the arthropoda and the cell wall of the fungi. So answer is the option D is the correct answer. So next question. Now, the equivalence of a thin cartilodon in monocots is scutellum. Is a scutellum. Now, next is the select the type of gland with appropriate example. Here, two options are correct. So, adrenal also an endocrine, also endocrine gland. This is also correct. Adrenal is an endocrine gland. Adrenals also endocrine gland. Adrenals is a endocrine gland. It's also correct. And mammary gland also apocrine type sweat gland. Mammary gland is a apocrine type gland. And thymus is not heterocrine. Heterocrine have those both have exocrine and endocrine function, like pancreas. Pancreas is a heterocrine gland. So pancreas have both exocrine as well as endocrine. Exocrines are those are exocrine glands. Exocrine glands, they produce enzyme. Actually, the measure of the enzyme producing glands are exocrine glands, and endocrines are hormone producing. So adrenals also, this should be both are correct. Adrenal glands is an endocrine gland, and mammary gland also is an apocrine type glands. Apocrine type glands where the uh, accumulates secretion, accumulates their secretory product on the at the apical surface of the secretory cells. So accumulates their secretory product at the apical surface of the secretory cells. Now next, so answer is both A and B. Now, the plasma membrane is the boundary between a cell's internal and external environment. Both environment are aqueous. Now, you know that there is the lipid bilayer, phospholipid bilayer is there, and the phospholipids are amphipathic in nature. Amphipathic phospholipid, it's have a hydrophilic hydrophilic head group hydrophilic head group and hydrophobic tail. Hydrophobic tail. And due to this presence of hydrophilic one molecule, both one end contain hydrophilic end, another contain hydrophobic end, then we called it as amphipathic molecule. So they are known as amphipathic molecule. Amphipathic molecule. The So phospholipid is an example of amphipathic molecule. So X is the region of hydrophilic layer of the outside, and these are hydrophobic region. So hydrophobic layer on the inside of the membrane. That's why phospholipid is known as amphipathic molecule. Phospholipid is known as amphipathic. Amphi means both. So hydrophilic in hydro means water, philia means loving species. Hydro means water and philia means loving species. So water and philia means loving species. So is fond of water, it's loving, that is hydrophilic in. Hydrophobic in is water hating in. So when a molecule contain both in, then we called it as a amphipathic molecule. So it's a answer is A is the correct answer. So next question. The botanical name of the plant source of colchicine is, we know that colchicine is obtained from a plant source known as Colchicum autumnale. Colchicine actually involved in the generation of polyploidy. It prevents microtubule polymerization and prevents to metaphase to anaphase transition that leads to polyploidy. It's a toxin that inhibits the microtubule polymerization. So that's why and it's important. So colchicine is obtained from Colchicum autumnale is the correct answer. So thank you.